All right, I'd like to welcome everyone to today's edition of the Polyploidy Webinar. Today we have two great speakers who are going to tell us about polyploidy and hybridization and different systems from yeast to plants today. Up first is Dr. Samina Neseeb. Samina originally grew up in Pakistan and completed her master's in microbiology at the University of Karachi, where she started out studying fungal xylonases. She then moved to the UK for a PhD in genetics at the University of Manchester, where she continued to work in fungi, studying molecular evolution in yeast, in particular the role of chromosomal inversions and translocations in speciation and adaptation. She's currently at Staffordshire University as a senior lecturer in molecular genetics and genetic engineering, where her work combines high throughput genome sequencing, phenotyping, transcriptomics, quantitative trait loci mapping, and other kinds of genetic analyses to understand the genetic basis of functionally relevant and industrially important traits in yeast. She's interested in understanding the role of non-coding RNAs in cell function and gene regulation as well, and studying the mechanisms required for evolutionary adaptations of organisms to environmental change more generally. Today, Samina is going to tell us about her work exploring genetic and phenotypic diversity of biotechnologically important yeast hybrids. With that, I'll let Samina take it away.
Thank you. Thank you, Mike, for the introduction. I hope everybody can hear me. So today I will be talking about exploring the genetic and phenotypic diversity of biotechnologically important yeast hybrids. This was the work that, as Mike mentioned, that I recently joined Staffordshire University. The work that I'm going to be presenting today was done when I was still at Manchester University, working as a research fellow in Professor Daniela Del Neri's lab. So just starting off with a brief introduction to yeasts. We know that yeasts are very widely distributed throughout all biomes, and most of the world's ecosystem in different communities of species, and they share similar ecosystem functions, properties, and interactions. They can be found from ripe fruits. They can be isolated from bark, roots, and leaves of different plants, deep soil, and insect guts. I am particularly interested about Saccharomyces sensostricto yeast species. When we talk about sensostricto group, people often equate with species Saccharomyces cerevisiae, which is baker's yeast and play a very important role, in production of fermented foods and beverages. However, the truth is that Saccharomyces cerevisiae, it represents only a tiny fragment of the vast diversity of the yeast world. To date, there have been huge progress in the discovery of new species, and now many yeast species have been discovered. Saccharomyces sensostricto group consists of 8 different species, which have been isolated from tree barks and surrounding soil, and from insect guts, and they have been found from from places all over the world. And this group of sensostricto yeast species also contains a natural hybrid, which is a hybrid between Saccharomyces cerevisiae and Eumayanus, and is known as Saccharomyces pastorianus or Carbalgenesis as well, and is very widely used in brewing industry. Saccharomyces uri has been the latest addition in the group of Saccharomyces sensostricto group, which was discovered by myself a couple of years ago from French Alps. And the recent studies have shown that it also plays a very important role in brewing. Yeast hybrids, they are not only restricted to Saccharomyces pastorianus and brewing, but they are found in other fermented beverages as well, in industrial applications, and also in natural environments. So hybrids between Saccharomyces cerevisiae and Paradoxus, they have been isolated from natural environments, from oak trees. Hybrids between Saccharomyces cerevisiae and Couturier zevi, they are very widely used in the wine industry. However, when we talk about yeast hybrids, the problem is that the interspecific hybridization, produces largely sterile offsprings, and there are many pre and post zygotic barriers that prevents the hybridization. And therefore, the successful hybrids, they are seen relatively rarely. So then why do the species hybridize? And it is because hybridization can actually result in production of novel genes and give us some novel phenotypic traits, which can be very important at industrial level. So it gives us an opportunity to combine it has an opportunity to combine the beneficial traits from both parents, and give, leading to adaptations that could not occur in either of the parental species. So in my today's talk, I will be focusing on yeast diploid and tetraploid hybrids. And the first part of my talk will focus on yeast diploid hybrids and the importance of the mitochondrial inheritance in those hybrids and how that inheritance of the mitochondria can impact on the fitness of the yeast species. The second part of my talk will concentrate on how we can overcome the problem of infertility in yeast hybrids and explore the traits that are beneficial for biotechnological purposes and understand the genetic variation that is actually responsible for different traits in using breeding process. So starting off from the first part about mitochondrial DNA inheritance, when we have the hybrids between interspecific hybrids between yeast species, when we made two different yeast species, the initial hybrid that is formed will have both copies of the nuclear genome from both species and mitochondrial genome as well. After about 20 generations, one of the mitochondrial genome would be lost. So the mitochondrial DNA in yeast hybrids is biparentally inherited. However, colonies after about 20 generations will rapidly lose one type of mitochondria and they will become homoplasmic, keeping mitochondria from one parent. Now, how does it decide that which mitochondria to inherit? It can depend on many different kind of factors. So it can be dependent on the temperature under the conditions in which the hybrid is being made. It can depend on the environmental conditions, like different media sources might have different kind of effect on the inheritance of the mitochondria. It can be dependent on the parental strains, other stress conditions, or maybe the inheritance of the mitochondria is random. So we set off by constructing the lab hybrid. So this work was done by an ex-PhD student in Daniela's lab, Sarah Hewitt. So what she did was she constructed hybrids between Saccharomyces cerevisiae and Saccharomyces euarum. So Saccharomyces cerevisiae is a thermotolerant species and can tolerate temperature as high as 37 degrees. Some strains can go even higher to 40 degrees. Saccharomyces euarum, on the other hand, is more cryotolerant and can grow at a temperature as low as 4 degrees. So it will give us a good system to understand whether the mitochondrial inheritance is dependent on temperature or not. So when we constructed the hybrids, as I said, they had the copies of nuclear genome from both parents, mitochondria from either of the parent, from either Saccharomyces cerevisiae and euarum. And the constructed hybrids can then be selected on selected media plate. And we can verify them by sequencing mitochondrial genes or by doing restriction digestion of different mitochondrial genes, just like over here we show for COX2. We can look at the restriction pattern of either the colonies are having for cerevisiae and euarum, and then identify which mitochondria our hybrids have inherited. So once we have constructed those hybrids, as I said, the hybrids were constructed at two different temperatures, 30 degrees and 10 degrees, so that it can kind of show the temperature effect for both species. And we also gave them different kind of media sources, which was a glucose-containing media, more fermentable condition, and a glycerol-containing media, more kind of respiratory conditions. And when we constructed the hybrids under those conditions, interestingly, we found that at a warmer temperature of 28 degrees, most of the hybrids that were constructed in glycerol-containing media preferably inherited the mitochondria from Saccharomyces cerevisiae compared to Saccharomyces euarum. And this effect became opposite when we constructed the hybrids at 10 degrees. At 10 degrees, as we know, Saccharomyces euarum is more like cryotolerant, and we saw that our hybrids, they preferred at lower temperature to inherit the mitochondria from Saccharomyces euarum in glucose-containing media. We couldn't construct many hybrids in glycerol-containing media, but the only hybrid, the successful hybrid we obtained, also had the euarum mitochondria. So overall, these results kind of showed us that indicated that Saccharomyces cerevisiae mitochondria is preferentially inherited over Saccharomyces euarum at warmer conditions, where respiratory ability is crucial, and vice versa. We then wanted to know whether the inheritance of a specific mitochondria is actually affecting the fitness of the hybrids as well or not. And we kept the conditions same. We tested the fitness in a fermentable sugar as well as in respiratory conditions. And we compared the fitness of our hybrids with Cerevisiae mitochondria and Euarum mitochondria compared to the parents. And as we can see, the fitness at 28 degrees was much higher for the hybrids which were having Cerevisiae mitochondria compared to the hybrids which were having Euarum mitochondria. Those grew much better at 4 degrees. This effect became even more prevalent and dominant when we tested the fitness in a non-fermentable sugar, which was glycerol. And as we can see in a non-fermentable sugar, we can clearly see that the Cerevisiae mitochondria are preferring to grow much better at a higher temperature of 28 degrees, whereas the hybrids with the Euarum mitochondria prefer to grow at a lower temperature. So these results showed us that inheritance of the mitochondria play a very important role in providing fitness to the hybrids, and can also have important role in giving important traits to the hybrids that are related to the temperature. So we wanted to understand what traits are being inherited, but what the problem that occurs with the diploid hybrids, as I said before, is that they are sterile. So if we want to study further generations and to find out which traits are being inherited and that are linked with the phenotype, we need to overcome their sterility. So this will lead to the second part of my talk, in which I will show you how we overcame the infertility by creating tetraploid intermediates of Saccharomyces interspecies hybrids, since a large part of industrial strains are hybrids. And we will see how we can use that natural phenotypic variation to improve the specific biotechnological traits in hybrids. So this project was a big project and was in collaboration with different academic partners and industrial partners, AB InBev, which was funded by BBSRC, in collaboration between Manchester University, Nottingham University, and Leicester University. So what we initially did was that we harnessed the biodiversity through hybridization by constructing many different kinds of tetraploids. We constructed, as shown in this figure, we constructed the tetraploids between Saccharomyces cerevisiae and Paradoxis, Cerevisiae and Uri, Cudrea zevi, Cerevisia and Uvina. So we tried to make as many possible combinations of hybrids as we can, so we can explore the diversity. And when we made the hybrids, we also took into account that we are taking very diverse populations of those strains. So for example, if we were making a hybrid between Saccharomyces cerevisiae and Cudrea zevi, we used Saccharomyces cerevisiae from North America and a Saki strain of Saccharomyces cerevisiae and a Portuguese strain of Saccharomyces Cudrea zevi and the Japanese strain of Saccharomyces Cudrea zevi, which really allowed us to mix the genetic diversity and to understand the traits that are being inherited and that are important for the biotechnological purposes. So I'll just show you how we did that. So to overcome the the problem of sterility in the hybrids, we constructed tetraploids. To construct the tetraploids, we first made hybrids between Saccharomyces cerevisiae, let's suppose it was from North America, and Saccharomyces kudryaszewi, which is from Japan. So we obtained the first interspecific diploid hybrid. We then made a second interspecific diploid hybrid between a different strain of Saccharomyces cerevisiae, which was Saki, and a different strain of kudryaszewi, which was from Portuguese. And we obtained a second interspecific diploid hybrids between Saccharomyces cerevisiae and kudryaszewi. Now, these interspecific diploid hybrids, they are sterile. To make a tetraploid from these, we deleted one met locus from each of the diploid hybrids and made them behave as a gametes. So our first interspecific hybrid is a diploid hybrid, but is behaving as a gamete with one mating type of mating type A. And the second hybrid is also an interspecific hybrid, but is behaving as a gamete with only one mating type, alpha. We then crossed these two interspecific hybrids to obtain a tetraploid hybrid, a fertile 4N hybrid. And at this step, we also selected for different kind of mitochondria according to the parental species we used. Once we obtained the fertile hybrid, we then sporulated it, and we obtained 2N hybrids, the progeny, which is now fertile, and we can further cross these progenies. We can let them grow, make them gametes, cross them, and repeat this cycle several times to obtain multiple generations of progenies, which are going to be fertile and can be then intercrossed with each other, can be sporulated, and can then bring another progeny from as a next generation. In this way, we did like 12 generations until we obtained the F12 progeny. We kept making the hybrids and sporulating it. Once we had made the hybrids, of course, we wanted to see if they are fertile, and they indeed were. We looked at the spore viability, as we can see highlighted in the red box. The tetraploid hybrids, they had a very high spore viability because they were fertile compared to the interspecific diploid hybrids, which were inviable. So after obtaining the hybrids, the first thing we asked was whether we can see any kind of variation in terms of fitness in our F1 progeny, as well as our last generation, that is the F12 generation. And whether we can see, like, because we know that now the hybrids would have inherited like traits from 4 different parents and will have a very diverse genome. So we wanted to see that if that diverse genome is going to give us any good fitness effects or not. This was done on a large scale using Singer HDA rotor in 384 well plates, and the fitness was tested under different conditions, again, like temperatures, stressors, and different kind of sugars. So we made sure that we are using the conditions that actually yeast is facing in our industries. And this is just an example of one hybrid. Obviously, we tested it for all the hybrids that we constructed, but for the lack of time, I'm just going to show you as an example for one of the hybrids, which is between Saccharomyces uri and Saccharomyces cerevisiae. We had hybrid with different mitotypes. So hybrid with cerevisiae mitotype, hybrid with uri mitotype, and hybrid with a recombinant mitochondria. And as we can see from this heat map, that already we could select some of kind of progenies which kind of outperformed compared to the parents, which was quite promising when we looked at the fitness of our F12 progeny. And it was even more prevalent, and we could see a huge distribution of our hybrids that we have, our spores that we had. They had a quite wide distribution of fitness, with some spores growing very, very low compared to the parents in terms of fitness, and the others having a very high fitness level compared to their parents. And again, we tested this fitness at different levels for all the different hybrids. But Now we wanted to know that, okay, that's good. We are getting the phenotypic diversity and we are getting the progenies, they are inheriting the good traits, but we want to know that what trait they are inheriting, what is the alleles that are kind of, related or linked to those particular traits. So for example, this is the condition for maltose, where we can see a huge distribution of our spore fitness. To understand the traits, we picked 20 low growing, very bad growing hybrids, 2N hybrids, and 20 best that were outperforming compared to the parents in terms of fitness, and we sequenced them. So we had 20 best progenies, 20 worst F12 progenies. We sequenced them, and then we did the QTL mapping to identify which phenotypic traits are associated with which genes. And we did that for 3 different conditions that we tested for fitness, which was low temperature, high maltose, and high acetic acid. And from our sequencing data, we made the QTL plots, as we can see over here in this figure. This is the x-axis is showing the chromosome coordinates, and the y-axis is representing the allele frequency. The red and the green lines, they are the allele frequencies for our high fitness and the low fitness spores. And the black line is showing the LOD score for the fitness, and then the gray area is the one that is representing the 90% confidence interval. So we can see that, if there is a very high peak over here for our LOD score, which was above than 10, it means that, this is significant, there is a significant QTL over here. So the first thing we asked was then about the mitochondria, because we know that mitochondria from our previous results can play an important role in terms of fitness. So we wanted to see if different types of mitochondria, they have an effect on the QTL landscape. And interestingly, so figure A over here is for the hybrids between Saccharomyces cerevisiae and Cudrea-Zevi. And B is for the hybrids between Saccharomyces cerevisiae and Juri. And interestingly, we saw that the majority of the QTLs deducted were exclusive to a specific mitotype. The yellow ones are the ones that are representing the shared mitochondria, are representing the shared between hybrids with different mitotypes, the QTLs that are shared between the hybrids between different mitotypes. And we can see, whereas the orange and the blue and orange and the green are the unique QTLs to specific hybrid. And we can see the majority of the QTLs are actually very unique to a specific mitochondria, and the shared ones are approximately 2%. So this reinforces the idea that mitochondrial nuclear interactions, they have a genome-wide effect, and they are important in context of evolution. To identify the relevant genes within the QTL regions, we investigated the overlapping QTL regions deducted in different hybrids. And so we took this approach because it can help us to identify unambiguously identify the genes that underlie the phenotypic effect observed. So for example, over here, for the acetic acid condition, we can see that there are shared QTLs between Saccharomyces juri and the Cerevisiae hybrid shared with Saccharomyces cerevisiae and Cudrea-Zevi hybrid, where, for example, NQM, is mapped in both of those hybrids, NQM, and it is a nuclear translocase, which is known to be induced by the acetic acid condition. So we can find the alleles which are specific to the certain conditions and are actually shared between different hybrids. So kind of identifying the causal genes. Similarly, we could see shared genes between different hybrids at low temperature. Over here, the COQ6 gene is related to the temperature effect. And then similarly, we can identify genes which are shared between different hybrids And over here, for example, MAL33, it is a causal gene and is involved in MAL activator protein, and it overlaps. So in this way, like, you know, the hybrids will it allowed us to focus on the genes that are more likely to be responsible for the phenotypic variation. We then wanted to understand if a particular gene, we wanted to validate the effect of our QTL alleles. So we chose 3 genes which were not causal genes, and we tested the fitness of allelic variants via reciprocal hemizygosity. So this is how we did the reciprocal hemizygosity. We created tetraploids by deleting our gene of interest. So first we made the diploids by deleting our gene of interest from one species and not from the other. So, and similarly between the hybrids, for example, Saccharomyces cerevisiae Saki and Saccharomyces cerevisiae Chinese. And the same thing we did for Saccharomyces cerevisiae North America and Saccharomyces cerevisiae Japanese. We crossed those interspecific hybrids. We got hemizygote tetraploid hybrids where one allele of our interest has been deleted. And we saw interesting results. We could see that, for example, in acetic acid condition, it was the ASI2 gene that that was deleted in our North American Saccharomyces cerevisiae and Saki Saccharomyces cerevisiae. And from this plot, we can see that the allele having the Saccharomyces North American deleted was outperforming in fitness compared to the Saki Saccharomyces cerevisiae allele. We saw the effects for first 3 and GIT1 as well, which are related to low temperature and maltose. And interestingly, we saw that by deleting those alleles, the fitness effects that we see overlaps with what QTLs mirrors with the effect that we were seeing in our QTL maps. So for example, this is the location for the first 3 allele, and this is what we saw, that, you know, on our QTL map, the green line is for the low fitness one, and which was the Saccharomyces North American allele, and the red line is for the high fitness one, which was for the Saccharomyces Saki. So the effect kind of mirrored what we were seeing in our QTL results. However, there was only this AC2 in which the effect didn't mirror. It was the opposite effect, and the effect in QTL was opposite as well. And it can have many reasons, because probably maybe the genes that are in the nearby proximity, they are masking the effects in the difference in the phenotypic screening, or also because the phenotypic screening of the F12, you know, that is done in the solid media, and whereas the QTL spores, they are grown in the liquid media. So other effects can also be taken into account. So we then validated, we wanted to know that if this effect that we are seeing is hybrid specific, or it is coming from just the diploids of Saccharomyces cerevisiae. So to validate and to, like, you know, have allelic variants were they exclusive to the interspecies hybrid background rather than to the intraspecies strain variation. So we did the hemizygosis analysis between cerevisiae diploids. Again, we deleted our gene of interest from one of the cerevisiae strain, and we made the hybrid. And then made a diploid hybrid between Saccharomyces cerevisiae North America and Saccharomyces cerevisiae Saki. And as we can see over here, when we made cerevisiae diploid hybrids, we didn't see much significant fitness difference. The only difference that we saw was in first 3, and that was also kind of opposite to what we were seeing in our tetraploid hybrid. So this is the diploid hybrid, where the North American allele is outperforming the Saki allele, whereas in our diploid, the Saki allele is outperforming the North American allele. So this kind of suggests that the allelic differences are influenced by the interspecies hybrid genomes. So just to summarize, we showed that the mitochondrial retention in hybrids is dependent on the environment. They can have significant fitness effects and can generate novel phenotypes. To understand those phenotypes and to understand the traits, we can generate tetraploid hybrids and restore the fertility, and then can understand the quantitative genetics that is related to the phenotypic diversity. We also showed that the species-specific mitochondria inherited in the hybrids had a very profound effect on the phenotypic variation. And new QTLs can be generated, which are specific to the hybrid background. With that, I would like to thank everybody, especially Professor Daniela Del Neri, in whose lab all this work was done. Without her support, it would have been very difficult. And of Federico Vicinoni, he did a great job with identifying all the QTLs and doing the hemizygosity analysis, all that part of the project. Sarah Hewitt did the mitochondrial inheritance part, and I would like to thank all the other members of Del Neri lab, all our academic partners, Professor Ed Lewis and his lab, Chris Powell and his lab, Professor Catherine Smart, and the genomic facilities, both in Manchester and in Arnhem Institute. This work was funded by BBSRC and AB InWeb. And of course, I want to thank the Saccharomyces yeast. Without them, I would not have been able to explore and understand all this genetic diversity. And I would also like to thank Staffordshire University, because I joined them in the middle of the pandemic last year in August, and they have been very supportive in helping me settling in and actually, establishing my career as an independent researcher. So thank you everyone for listening.
All right. Well, thank you very much, Samina. That was wonderful. We're just at time, so we have maybe a minute or two for a really quick question before we switch over to Eric's talk. So if anybody has any questions, feel free to pop those in the chat box or turn on your audio, video, and fire away.
Will I be able to see the chat box?
Maybe.
I can't see any option for the chat box, so maybe if someone asks the question, you can just shout out. Chat here. Okay, I can see the chat.
I had a real quick question before we switch over too. So it seemed like there's a lot of the variation in the phenotypes of the yeast, depending on not only the hybridization status, but also which mitochondria they have, and as well as ploidy level. So it seemed though that maybe mitochondria had a larger impact on these traits than ploidy level itself. Is that what I'm reading correctly? The mitochondria itself. Mm-hmm. How different though are those mitochondria between the species? Do they have the same? I assume they have the same core genes. So this is sequence divergence that seems to be driving that difference, or are there different copy numbers on the mitochondria between the species? Yeah.
I mean, the copy number of the mitochondria can vary as well, but they do have different sequences as well. So they have the sequence divergence. So really the effect can be driven by both of those. Of course, we haven't yet pinpointed exactly whether the copy number is also playing any kind of role, but yeah, both of them can have the effect.
Right. No, that was very, very cool. All right. Well, if there are any questions for Samina, we'll have to take those at the end after both talks. So save those, and we'll go ahead and let Eric share his screen and get on to the next talk. All right. All right. Well, up next is Dr. Eric Schranz. Eric is the head of the biosystematics group at the University of Wageningen. Hopefully I Wageningen correct this time. Close. Eric's lab studies the evolution of plant diversity and genomics, and in the last few years has done some really, really cool work, I think, on the understanding of the phylogenetic analysis of synteny. And today is going to tell us about his work on the tetraploidy to hexaploidy, the ploidy two-step in the Cleomaceae. With that, I'll let Eric take it away.
Thanks, Mike. Can you hear me okay? Now I don't see you guys.
We hear you loud and clear.
Okay. I'm not sure where the pointer is, though. I feel like an old man. There it is. Automatic. Okay, do you see? Oh no, that's my laser color. Okay, yep. Yeah, great. Thanks so much, Mike. It's fun to talk, and especially with this group, and to be able to talk to my American colleagues. Of course, also my international ones. But I absolutely feel kind of stuck in my little bubble in the Netherlands. So it's fun to talk to people on Mountain Time, because I'm originally from Colorado. So I appreciate the chance to do so. So there's a few jokes in here that are meant for my American colleagues, and I apologize for that. You have to make your life fun somehow during these times. Indeed, I kind of struggled, should I talk about the synteny stuff, but I've been talking mostly about that. So I actually am returning back to kind of where I got my start, and I also wanted to give this talk about Cleomaceae, because this also connects me and Mike and a lot of the colleagues that I thought would be talking here. So it's kind of a reflection on where we've been over the last, God, 20 years now, which makes me feel old. And where still exciting things are going. So maybe for the young students, that's also kind of fun just to think like, yeah, when I was a young PhD student postdoc, I'm still working on the questions that have been interesting me basically my entire career. And that's kind of satisfying too, to look back and start thinking like, you know, there's always new questions that pop out of the things that you do now. So indeed, I think partially I came up with this name thinking about dancing a little bit, the two-step, and I think it's partially because we tend to think about the importance of tetraploids and the interaction of two genomes together, but we all know there are plenty of great examples of three genomes interacting, right? And I'll introduce those in a second. And certainly this crowd, I don't have to even say these things, but just to remind ourselves, there's lots of great information about the extent of recent polyploidy across the globe. These are for plants, but clearly many other lineages such as insects or yeast that we just heard the nice talk about. And many of these recent polyploids are important for us, such as foods. And we all know, for example, with the 1001 transcriptomes and long before that, how ubiquitous ancient polyploidy is. So all the red colors in this phylogenetic circle show history of some sort of ancient polyploidy. And so clearly I put a fern behind me for Mike there to remind us, you know, there's lots of ancient polyploid ferns. We see it now in a lot of gymnosperms, but widespread across the angiosperms. So even though we see so much of it though, right, there's still this question, we don't see quite as much ancient polyploid as you might expect from extant polyploids. And this gets back to this question of, you know, there might be a short-term benefit to polyploidy, but it might run out and lots of things go extinct. So how do you solve that problem of kind of the initial benefit, and then you've got to diploidize, fractionate, and all those things to stabilize and actually become a successful ancient polyploid? And that's something I know many of you guys, Mike included and myself, have been quite interested in for a very long time. And so when we think about polyploids, right, and one of the issues is there's many flavors of polyploids. So this is kind of a retro sign from when I was growing up, Baskin-Robbins and their 31 flavors of ice cream, right? And so when we think about polyploids, I think we all know this, that we can either split it up by the numbers of chromosomes. So if we call it talking about triploids or tetraploids or hexaploids or octoploids, but we tend to also split it into the typical flavors of auto versus aloe. And it's important to remember that there's every combination kind of in between there, right? There might be segmental Aloe Polyploids or even aneuploids that come out, or chromosome breaks, things like that. So it's not black and white. There's not just auto aloe, and there's not just probably triploids, tetraploids, hexaploids. There's some sort of mix. And I think that's what's quite interesting. And like I said at the beginning, I think when we look especially at extant polyploids, we tend to think of there being lots of tetraploid examples out there. So maybe it's like the chocolate chip flavor. And hexaploids, are they a little bit more rare? I don't know. That's actually something I'd be curious to hear from everybody in the audience if they think so. So can we think of tetraploids as kind of the standard ice cream you get every day, the generic chocolate chip? I don't know what's more generic than that. I just wanted it 2 flavors. And then our hexaploids, kind of the more special Ben & Jerry's flavor of the day, some sort of weird combination of things that you would never think should be mixed together. And so I think all of us are fascinated by the possibility of aloe hexaploids, and especially because of all the beautiful papers over the last few years on wheat, right? And wheat is kind of the poster child of a good Aloe Polyploid with our well worked out model of the A and B genome coming together first to give rise to the AAB species, bring in a third genome and get this, the modern day bread wheat, and each of the chromosomes still behaves quite well. And I'm just curious, I don't know if I can ask a question to people, but can anybody name for me another good Aloe hexaploid recent crop? Anyone? That's a good test if you guys are actually listening to me too.
How about Saccharomyces? I mean, not saccharum.
Yeah, well, what is, sugarcane? I don't know. Is that even a hexaploid? That's some sort of weird beast of a genome, right?
Okay.
I think it's even more. It's like a dectaploid with all sorts of aneuploids and stuff in it. But this is good. I actually struggled a little bit myself to come up with something besides wheat that was a good Aloe hexaploid crop.
Just so you know, Eric, you can't see the chat box maybe, but we got putting in camelina and oats and strawberry, all sorts of stuff.
Camelina is a good one. Yeah, oats is the other one. Indeed, that's true. Yep, good. Okay, indeed, those are good ones. So people are reading the literature, paying attention. Oop, now where am I? Okay, and another two that I came up with personally were kiwi is also turns out to be an Aloe hexaploid, and although not a eating crop, an interesting horticultural crop is the chrysanthemum. And so this was actually a PhD defense I was part of not too long ago, disentangling hexaploid genetics. So, but indeed, I think compared to the number of tetraploids, we have fewer kind of role model recent Aloe hexaploids, which begs the question, well, what is it? And indeed, when we look back at the tree, and obviously this is a little bit of an outdated one from Koji, but when we plot on here the number of ancient tetraploides or doublings, you see quite a few of them. But when we look at the number of ancient hexaploides on here, there aren't nearly as many as we see tetraploides, but there are some really interesting and super, super important ancient hexaploids, right? So for example, here at the origin of eudicots, the gamma event at the origin of the Solanaceae, also the Astraceae has one. And then of course, for example, Brassica has one, and I'll talk also about Cleomaceae in a minute. So these two that are kind of close to each other is the point that I want to talk about. So this is just to remind us, grape, this was a huge deal when it came out, finding the triplicated regions due to an ancient hexaploidy that's now shared by most rosids and asterids. I did some work when we sequenced the petunia genome, comparing it to tomato, for example, and we could actually show that indeed petunia is also triplicated, but has fractionated a little bit differently than the other traditional Solanaceae, such as tomato or potato or Nicotiana or capsicum. And some work that Mike did, I think that was quite nice, and this was a 2016 where he actually showed, first they had found that the core compositae shared a hexaploidy, but then Mike went in with some phylogenomic analysis and could actually show that, for example, the Barnesoidy is a tetraploid, and then you get the addition of another third genome much later in time, giving rise to the bigger radiation. So I think these are nice examples of potentially how you go through different steps. I see there is a question, so if anybody wants to speak up, that's also okay. Mike, you can also interrupt me. But like I said, those are kind of some examples, but then what I really like is the idea of within the Brassicaceae and its sister family, the Cleomaceae, a group that I've been interested in for a long time, you actually have two hexaploides. Well, and then we also have camelina. That's a good example. I should put that in here. But such that the genome triplication in the Brassica lineage actually happened after the alpha. So it's a duplication followed by a triplication, giving rise to essentially a 6x genome. Whereas over here, we have a 3x genome. And I'll get into why now. I think it's actually a 2x followed by a 1x. So a little bit more like the compositae example rather than a single step. Okay. And why I like these two families, and everybody knows the Brassicaceae for the crop Brassicas and Arabidopsis. Its flowers are not very particularly exciting and interesting, as the name suggests, crucifery, 4 petals, 6 stamens. Pollinators are not that diverse, but they go absolutely crazy in terms of fruit morphology. And distribution-wise, they're particularly in the temperate regions, rarely in the tropical regions, as you see here, whereas its sister family, the Cleomaceae, is kind of the opposite on all of these characters. Much more showy, zygomorphic flowers, much more diverse pollinators. However, its fruits are really boring, basically just these simple fruits like this, and its distribution is mostly in the tropics and the desert regions of the world. So, and you see this pattern a lot of a temperate family sister to a more tropical and desert family. So it's also kind of a nice example of that. Okay, so what, with this nice system and these different ploidy levels in the Brassicaceae versus Cleomaceae and different genomic ones, then my general question that I've been interested in for a long time is basically, what's the role of genome duplication and genome repatterning, genome retention and loss, or fractionation? How does this contribute to networks and, for example, diversification? And so this involves these polyploidization cycles followed by diplization and loss. And does this potentially drive diversification? And specifically, then what are the consequences of going through a tetraploidy followed by an additional one, such up to hexaploidy, for example? Okay, and I already mentioned this before, but the other players I didn't introduce here that are kind of interesting and important is we also, work I've done in the past has been to do the genome of Aphionema, which is quite interesting because it's basically the first lineage immediately after the alpha event. And clearly diverged from the rest of the core Brassicaceae. Part of the BMAP project, along with Chris Pierce, for example, we've done the genome of Cleomaceae violaceae. Oh, sorry, there's an extra E there that needs to go away. And it went through neither the alpha event nor these polyploidy events later up in the Cleomaceae. So it's basically like the 1x. So you can see these are the kind of essentially genome contributions that you have across the system, which makes it, I think, quite a powerful, interesting system. Okay, so, and as somebody once said, one's company, two's a crowd, and three's a party. So that's one of the things we can maybe look at in here. Okay, and this is where I get kind of a rear view mirror look at what's happened over the last while. And actually, I really have to thank Mackenzie Marbury. She just recently defended her PhD defense with Chris Pierce, and now is working with the Soltices. And it was really nice. Her thesis was kind of on looking with transcriptomics at where polyploidy happened across the Brassicales. And in her thesis defense, she kind of did this nice summary of these key papers, which also made me feel nice that somebody is following up on some of my earlier work, but also made me feel old, realizing it goes back to now 2006. So I just want to go through a little bit of this because I realize some of you may not have ever heard of these papers. But for example, in 2006, this was way back when back libraries was the best you could do. And we actually went in and looked at a region that was well known to be duplicated in Arabidopsis. So this involved the constans gene, for example. And we went into our Terrania library that we had just built, and we were able to pick out 3 regions that corresponded to the 2 regions in Arabidopsis. And we could actually show, based on KS values, that it was clear that the duplication in Brassicaceae was different than the apparent genome triplication in the Cleomaceae. So we got a paper out of that just by analyzing these back regions and the kind of analysis of genome fractionation in these. And then later, this was quite nice. I went to a meeting and met Mike Barker when Mike was still a pup, right, doing his PhD in Indiana. And we got to talking about his dupe pipes analysis. And I said, oh, I have this transcriptome of this Cleomaceae. I'd love to confirm what we saw with the backs. Could you help me out? And then we published this paper in 2009. And I think, Mike, this is correct. This was only your second paper that was not on ferns during your PhD. So this was also quite nice that we got a nice paper out together way back in ancient history now. And indeed, this shows then the KS values in the analysis where we could show, for example, in Brassicaceae, so the typical AT alpha has a KS value at about 0.8, and you see this kind of rise in the peaks. Whereas then Terrania, we saw a peak at a much younger age at about 0.4 that was clearly independent of this one happening in the Brassicaceae, whereas they shared the beta and gamma events that were older. So yeah, just to show us this, I wish I had an older picture from way back then, but this was the more recent one. Okay, and then later what we did is we then had a transcriptome of additional Cleomaceae, which was Gynandropsis gynandra. And I show it here because this was kind of a fun project to get into, because it turns out Gynandropsis is closely related to Terrania, but Terrania is a C3 plant, whereas Gynandropsis is evolved C4. So it's the closest C4 plant to Arabidopsis. And we were curious, did this polyploidy event potentially contribute to the evolution of C4-ness within this? So we were able to go in then and look at the transcriptome that some of my colleagues, including Julian Hibbard and Andreas Weber, for example, big people very active in the C4 field. And when we lined up the KS plots of Terrania and Gynandropsis, we could basically see, indeed, that they seem to share this peak. The duplicates were basically shared in common, and many of the genes that were duplicated in one were duplicated in the other. And looked at, for example, its potential role in C4. And we were kind of scratching our head because it actually seemed like C4 related genes, there were more C4 related duplicates in Terrania than in Gynandropsis, even though Gynandropsis is the C4 plant. So that was where some of the mystery started to begin. And then later we did the genome of Terrania. So, and this was just what we had already seen before with transcriptomics and with those backs, and basically just to show that indeed we could nail that. Here was the divergence between Brassicaceae and Cleomaceae, followed by the Brassicaceae specific AT alpha, followed by the Terrania genome triplication. And just for comparison, we also threw in Brassica. And this was kind of nice because Brassica is also a genome triplication, but we could show that it happened almost at around the same time that Terrania did. So in two lineages, we have two genome triplications then of approximately the same age, and thought, oh, this could be quite an interesting system to compare all these. And so we dug into, for example, this independent genome triplications in Brassica versus Terrania. And this is just an example of, for example, we looked at the Madsbox family. Key regulators, and we put where these different gene families duplicated, whether it be due to alpha or specific to Brassica or Terrania. And what was quite interesting, by just kind of doing this across all of it, we could show Arabidopsis for Madsbox was about 25% retention in the Madsbox, which is about equivalent to the genome-wide average. Terrania just had a few more, even though it's actually a triplication versus the duplication, 29%. But compared to Brassica, which actually has retained almost 56% of its Madsbox genes. So this was kind of interesting, and maybe, well, I'll come back to that at the end of my talk, in fact. Okay, so potentially this somewhat contributes to the diversity in phenotypes that you particularly see in the Brassicas, right? And this is something that Mike and I know just published a paper on diversity in genes related to the ancient polyploids in the diploid brassicas that potentially still contribute to phenotypic divergence within them. And then more recently, I wrote a review article with one of my longtime collaborators, Jocelyn Hall, and also Eric Rolson, the small Cleomaceae community, where we just kind of summarized the overall phylogenetic relationships and its worldwide distribution. And just to remind you then that we have Terrania up here, and for example, Gynandropsis here, and they're kind of considered to be up in this clade that's mostly in the New World or in Africa, for example. But you can see this TH alpha where we still don't know exactly where it is. So this was kind of the starting point then for Mackenzie's PhD thesis, or one of the questions she wanted to do, and was published just last year in AJB. And this is just to look at the different species she's looking at. But again, here we have Terrania, here we have Gynandropsis, for example. And she went through and started to look at where we thought polyploidy happened. And these were the two that I talked about before. I told you before, Cleomaceae violacea has no evidence of that. We know that. And then she used several different methods of looking at transcriptomes to look for evidence of these little middle peaks of these humps of polyploidy. And just to summarize that, then this is what she found, is that within this entire clade, it looked like there was support for ancient polyploids. This one, I can't remember the reason why we don't see it here, but potentially then it's interesting to look at where do all these peaks line up. So now we'll go through a whole bunch of slides very quickly, which is just to show then, for example, the divergence between M. giganticus and Terrania. So we can see a peak there. We can do Gynandropsis versus Terrania, for example, which is kind of the comparison we looked at before. We can look at other sorts of species, for example, one step further back, right? And we can start adding all these up, and you can basically see evidence of rapid speciation basically within this clade, such that the KS values are basically all somewhat in line, except for with a few exceptions of the closer ones. And of course, there's some differences in life history, so these peaks are not perfectly aligned, for example. And then when we look at the KS values, you can see then this hump here where it seems like everybody's lining up. And indeed, so this is what we think is potentially this TH alpha event. And what's going on here then is this then, are all these triplicated or not? And so that's then the next step in the picture is then we did the genome of Gynandropsis and analyzed this Cleomaceae violacea, which was part of this BMAP sequencing project. Okay, and the reason we did Gynandropsis was not only because of the evolution of C4, but also we got some money for it because it's actually considered a crop in Africa and Asia. So it's a leafy green that you eat like spinach, especially in Africa, and it's often pickled in Asia. And so this was quite nice to get involved with a big group that was interested in these leafy greens and orphan crops in Africa. And so again, we did it because we also observed variation between these Asian and African lines in terms of C4 related traits, and I won't get into that now. We also did a lot of analysis of worldwide germplasm pool. So we had about 96 lines, which represented West Africa, East Africa, and Asia. And when we looked at a number of different important characteristics for breeding and for vitamin content, for example, tocopherols versus carotenoids, for example, we could easily show three kind of distinct germplasm pools. So we were quite interested in looking at this in more detail. So we did a genome of this, and I'm not going to get into busy numbers of genome assemblies, but we initially did a Illumina plus 10x assembly, had a bunch of little scaffolds, not the best N50. And then more recently, we just got back some a high-rise assembly using dovetail ligation, and we're able to then collapse this into many fewer scaffolds, still not with a perfect chromosome level assembly, but about 41 MB. And an N90 you can see has really gone up a lot. So now for looking at kind of comparative genomics, we can do a lot more. This is just to show, indeed, we basically got 17 nice, nearly, well, there's actually 17 chromosomes, and we had basically 17 large scaffolds. So I just said that we didn't have chromosome level assembly, but we're actually, I'm wrong, we actually are quite close to getting chromosome level assemblies for this Gynandropsis, and that allows us to look in more detail at, for example, what is the nature of this polyploidy event. So I already told you we knew that Terrania was genome triplication, and now we're comparing Terrania versus this Cleomaceae violacea, which we suspect has no polyploidy in it. And indeed, if you line these up, so even if you don't stare at these all day, you can kind of see that there would be on violaceas on this axis, Terrania on this axis, and you can see 1, 2, 3, 1, 2, 3. And if you kind of scan across the whole genome, you find the vast majority of things in 1 copy in Cleomaceae violacea, 3 copies in Terrania, which is what we expected to see. And here's again the KS values. But to my great surprise, then when we looked at our friend Gynandropsis and started analyzing it, we saw a very different pattern, which was instead of everything being in 3s, we saw everything in 2s. And I think that's quite easy to see these blocks of 2 as opposed to 3. However, the KS values are still about the same. We zoomed in into particular regions, that back region that I showed you before, and just to show you, for example, this would be one particular region in Arabidopsis. Here it is again. And what we could basically show is that we had 2 things in Arabidopsis thaliana, 2 in Athionema, 1 in Violacea, 2 in Gynandropsis, and 3 in Haslerena. So exactly what we saw. So just to summarize, 2x in Brassicaceae, 1 in Violacea, 2 in Gynandropsis, and 3 in Terrania. So this kind of cool, interesting pattern. So what do you do when you have a whole genome like this? You can stare at it, start looking at different genes. We haven't gotten all that far in it yet, but one thing I was quite curious in is looking at the patterns of fractionation. So this is just the idea that will you see a difference between the 2 copies in terms of the degree of fractionation between them? So this would be then what most people would say, for example, the least fractionated and the more fractionated version of it across a fairly large window relative to Violacea, and this would be scaffold 5 and scaffold 1. And so you can see sometimes there's a difference of fractionation, sometimes a little bit less, but notice that this is up at around 80% of the genes in Terrania, and Violacea are still present in this copy of Gynandropsis, whereas here we're down at about 50. We compare these exact same two regions to Terrania now, and then you can see on average we get 3. It's maybe easier to see down here. We have 1, 2, 3 regions, but even the least fractionated one, we're down at about 50% retention. So compare 50 versus 80. So even though they seem to have a shared potentially tetraploidy step, the amount of fractionation of them is quite different. And even the slightly less fractionated one is quite high compared to these over here. So I think this is a really cool example of a potentially shared polyploidy event or partial polyploidy event, whereas fractionation has done something quite different after it. And of course, then investigating what kinds of genes that is will be quite interesting. And indeed, I come up then to a paper not from me, but out of Hong Ma's group and his co-workers, where they looked at all of the available genomes and verified whole genome duplications, and they plotted here the percentage of retained genes after a polyploidy event and the synonymous substitution rate, right, which is this kind of clock. And so, for example, Brassica rapa is actually a little bit below average in terms of its percent retention, which is kind of what well, it's not what I just said, but you can see it's down here, whereas Terrania was already considered to be actually one of the most highly retained numbers of duplicates. And I just showed you Gynandropsis even seems to have more retention. So I think this is quite cool that I don't know where Gynandropsis will be, but I would expect Gynandropsis will be somewhere up here, really at the high end of retained duplicates at this fairly mesoploid type KS value. So I'm quite interested to plot and look at this, and clearly quite different between the two Cleomaceae species, probably compared to, for example, Brassica, which is kind of down here. Okay. And what I find quite interesting is, so on the one hand, I just said that it looks like Terrania has fairly high retention of overall genes, and Gynandropsis I will think is even higher. But if I remind you what I said here, is actually Brassica for Madsbox genes was the high one, and Terrania was lower. So I think this is quite interesting too, coming back to that paper that, for example, Mike just recently published in New Phytologist on the role of genes retained after duplication and their role in domestication. And I think things probably like these Madsbox genes that are so key for phenotypic evolution are great examples of probably the types of genes that have been maintained in Brassica at a higher rate, whereas Terrania on average has retained much more, but clearly not of this particular family. So I think there's a lot of cool stuff that we can start doing with this, and some really interesting questions then to bring it full circle then. How do we look at this two-step of polyploidy in the Cleomaceae? So the 2 plus the 1 here. And I know that people, Sean Abrams, for example, I know is working in his PhD, also working with Chris Pierce to identify the hopeful tetraploidy ancestor of the Brassica hexaploids. So I think that would be super cool if we can actually find in the Brassica clade essentially the equivalent of this, and then directly compare those two and fill in the different gaps of these different steps in the polyploidy. And so what's the take home? I said, oh, are the hexaploids the cool special one? But now I just showed you actually Gynandropsis, and every species is cool, right? Every species has survived. But in some ways, Gynandropsis is often considered to be extra cool and special because of the evolution of C4, or at least I think it is. So it just kind of hammers home the question that, like, you know what, polyploid happens, lots of stuff happens, genes change, and maybe it doesn't always lead to new innovations, that every single one of these is some sort of interesting ice cream flavor. Some of them might be weirder flavors than others, but if you love ice cream, then you should love all the plants that are out there, whether they're polyploids or not. And so with that, I want to thank all my collaborators. I list most of them here, and here's my group in Wageningen standing in front of a sea of Terranias, and with this orphan crop consortium sitting in a field of Gynandropsis in Benin. And thank you then for all of you for listening. And again, as Andy Warhol once said, one's company, two's a crowd, and three's a party, perhaps. And I'm happy now to take any questions. And I'll stop sharing so I can see you all. And I think it was okay on time too.
All right, yes, thank you. That was awesome, Eric. As always, if there are any questions, feel free to pop those in the chat box or pop on your audio and video. I have a real quick question while we wait on everybody else to type their stuff in. You know, we recently sort of analyzed a lot of the sort of fractionation rates, and or at least the amount of genes retained in duplicate or triplicate. You know, and found that there wasn't a strong correlation with time. That, you know, once you count for the phylogeny, it was wasn't a very strong relationship. And we've been sort of trying to tease apart that you know, what drives this rate of duplication. And one of the things that we've been thinking about, that things that might drive the rate of gene loss may be mating systems. So do you have a sense of the differences in mating systems between among those species?
Well,
like, I don't know enough about Gynandropsis to have any idea about it.
No, that would be a cool idea. And actually, I should almost want to put in a slide in here, because we also worked a little bit on the genome of Castanospermum, which is kind of at the base of the ancient Polyploidy in the legumes. And it almost fractionates, it retains like 85% of its duplicates.
Yeah.
So it is total, but that makes sense because it's a tree. It's a tropical tree, so you'd expect
so it's pretty slow.
Whereas Gynandropsis, that's what I find really interesting. Gynandropsis is a perfectly good annual. Basically, it's not that different than Terrania. It's also outcrossing. Both kind of do this weird mix of outcrossing and selfing. They actually can turn their male and females off. So there's not a clear signature.
One of the students in the lab, Michael McKibben, one of the PhD students in the lab, has been working on this and following up sort of and thank you for plugging our recent new phytologist paper, but following that up across many other crops. And, you know, the rate of fractionation seems to be really important. So we'll connect with you later about some of that too. There should be some fun stuff there. Anyway, I see other people turning on and raising their hands. I think John's up next.
Hi all, John Lovell from Hudson Alpha here. Eric, great talk.
Nice to meet you, John.
Actually, really awesome complementation of talks today. I wanted to build on Mike's question and just ask how you think maybe like the very first few generations after a whole genome duplication event might proceed kind of like the mitochondria of the yeast, and how maybe being a tree or being able to self and life history might play into the patterns of fractionation, because you might imagine that a lot of the fractionation that we observe in extant populations really happened within a few generations of the whole genome duplication event. I wanted to hear what you might have to say about that.
Yeah, no, I think you're probably very right. And that gets back to the flavor question too. And right, there's been this debate in the literature for a long time that autos versus allos are going to fractionate differently. And I think everything we've learned about polyploids, right, in the last few years is some polyploids are really heavily methylated immediately, and there's lots of immediate dominance, and some are not. And probably that initial dominance has so much to do with it. And I think the divergence between it, right, we see some in Arabidopsis crops where, you know, if you cross Thaliana and Arenosa, right, or is that wrong? Lyrata, then you see immediate massive dominance of one over the other. So you can imagine how that might really drive massive fractionation. Maybe like in something like Brassica that happened, and whatever the two Cleomaceae were, Terrania and Gynandropsis ancestors, were not super diverged, but probably still allo, but not so diverged that then one was clearly the winner. So you could actually have this kind of medium degree of fractionation, and whoever this third genome was that piled on, I don't know, if it was more dominant, it screwed all that up. I just think of basically after polyploids, it's like being in a blender, right? The more I look at things, like sometimes it's clear, and sometimes it's like you just turn the blender on and shit happens, polyploid happens, right?
Thank you.
Yep.
Eric, I just want to add to that comment. When I was interviewing for my PhD with programs, that's what Lauren Reisberg told me when I wanted to study patterns of gene evolution in poly. This is back in 2003, 2002. And he just said, I just think shit happens, Mike. And so maybe we haven't come very far. I don't know. I always say, I want to know why that should happen. That was
Why should happen? Right.
Anyway, I think Brittany's up next.
Actually, I think Wilhelm had a question before me.
Absolutely. There you go. Yep. Wilhelm, if you want to go ahead and pop on.
Sure. I just wondered why you expected the Gynandropsis to have higher duplicate retention. You were mentioning that you expect it to be very high. But,
because of that fraction. Do you remember that fractionation graph I showed? I went kind of quickly through it, but basically what that fractionation graph is showing that we have one genome of Gynandropsis, where one subgenome that retains almost 80% of the genes that are found in the single copy ancestor. So it's clear that genome wide that at least one of the genomes is quite heavily retained. And the other one, even the least fractionated one, was still around 50 or 60. So if you combine those two, that's what, like say a 75% gene retention. And this gets back a little bit why we were confused when we did that initial transcriptome analysis of Gynandropsis and Terrania. We saw a lot of the same amounts of gene duplicates. And so we just assumed they share the hexaploidy. And I never would have thought, and that's maybe another lesson, like I'm not afraid to admit I'm wrong, but that was like, we just assumed it had to be the same. And now I never would have guessed that until we did the genome, that it was basically one was a tetraploid, one was a hexaploid, but that the hexaploid fractionated so much that basically the total gene copy numbers look about the same. Right, so that's also one of those things. Like sometimes you look at your data in a new light and realize that you were wrong once upon a time. So, does that explain it a little bit?
Yeah.
Brittany.
So first, thanks for a great talk. That was awesome. And this question kind of piggybacks on what John was asking earlier, but I know there was a paper that just came out a couple of weeks ago in, I think, the Schranz lab, I'm probably butchering that, on Senecio carneologous and looking at production of 5X hybrids between sympatric tetraploids and hexaploids. And the 5Xs retain some degree of fertility, and the aneuploids also retain some degree of both fertility and viability. And we've seen a similar thing in Campanula, and I'm wondering if, you know, early on in the origin of some of these hexaploids, while they're still in close contact with their, you know, tetraploid progenitors, you know, are we getting some sort of, you know, gene flow between the two that's causing what looks like gene loss until they sort of settle out into their own, you know, stabilized lineage?
Oh, that's interesting. Yeah, I don't know. I haven't read the paper, so I can't say. But the only thing that always does blow me away is just the way that, you know, certain lineages clearly do have an upper or lower limit to their ploidies. And I think that's always interesting that ones, and John, who asked a question earlier, and we both worked on Bucra for a while. And, you know, Bucra is a perfectly good crucifer genus where you see lots of crazy polyploid, but you almost never see anything above triploidy. You see one or two tetraploids come up in the wild populations, but they don't stabilize. Whereas, you know, plenty of other lineages love to be crazy high ploidies.
Yeah.
So I don't know why. It's same thing with chromosome numbers, right? And lots of people look at chromosome numbers. Some plant families like the Solanaceae I was talking about, lots of chromosome breaks, multiple polyploids, but it loves the chromosome number like I think it's 8. Or 9. It loves stabilize, or no, that's Brassicaceae. 11. Sorry, it's 11. But so why things have these upper limits, lower limits, I don't know. So maybe 5X is perfectly good for Senecio, but 4X is not good. I don't know. But I also want to say I really enjoyed the previous talk too. Maybe give a shout out because I love the yeast work in general. There's so much power you can do that, and I love what you were doing with the cytoplasmic nuclear incompatibilities. And I think anybody also needs to go back and read those early papers on, you know, I think that's one of the best examples of where fractionation followed like these observable rules, where that first few lineages after the Saccharomyces fractionate in a completely different way than the crown group, and it kind of ticks up in predictability as you go up it. And I wish that that was true in plants. Maybe we'll find one. Maybe the grasses is a good one for that. I don't know.
Yeah, good point. It was a nice talk.
Thank you.
All right. Are there any other questions? Yeah, for Eric or for Samina? All right. Well, if not,
see, now it's getting dark here. Finally.
There you go. If not, those are two great talks, and we'll post the recordings later today. Otherwise, I just want to highlight everybody to the talks, the next Polyploid webinar, which will be in 2 weeks. Those two talks will be from Wendy Van Drunen on the impact of urbanization on polyploid versus diploid plants in Canada's largest metropolitan area, and Shelley Gaynor on modeling the dynamics of multiple origins in gene flow and autopolyploids in the diploid progenitors. So stay tuned for those two talks in 2 weeks, and hope everyone has a good evening. And again, thank you to both Eric and Samina for 2 excellent talks today.
Thank you. Thank you very much.
Good afternoon and good evening, depending where you are.
Thanks.
Okay, bye.
Take care. Bye.
Bye-bye. Bye-bye.